Okay, so I'd like to first start by thanking the Junior Investigator Network for allowing me to present and share my past and ongoing work. Hopefully it's going to be interesting to a larger audience, and my work is centered on obesity and endothelial dysfunction. And specifically, I'll be sharing my work that's interested in this flow-sensitive, inwardly rectifying potassium channel and distinct vascular beds. And again, my name is Fancher. I'm an assistant professor in the Department of Kinesiology and Applied Physiology. Jumping right into obesity as a major health problem. And we usually associate going from maybe a lean, healthy individual over time with maybe a poor Western diet and industrialized nations and a sedentary lifestyle, lack of movement and sitting around, and in combination with a poor diet. We're also now understanding that there's a genetic component to this, but really the interventions are being targeted here in terms of diet and lifestyle. And what's kind of scary now is that obesity has achieved the status of global epidemic. And so this is a worldwide problem. And the study from about 10 years ago extrapolated this out to the year 2030, and estimates from this study suggested that there's going to be a 20-fold increase. And 10 years later from this study, this number unfortunately has not budged. In more recent estimates, we're still thinking that there's going to be over half the world's population will be obese by this year. And so this is something we need to prevent. And a major risk associated with this is cardiovascular disease. And so preventing this risk factor alone would help improve both economic and health modalities of obese individuals. Unfortunately, these specific pathophysiological mechanisms, or especially with the vascular disease, are poorly understood. And so that's where I come in. And so one of the hallmarks of vascular or endothelial dysfunction is flow-mediated vasodilation. Now, this is a shear drag force on the wall, the endothelium, the single cell layer of an artery that produces this laminar flow profile, and it induces a shear force on the vessel wall, which results in a mechanotransduction of the mechanical stimulus into chemical signaling, and that results in vasodilation. So this frictional shear force induces a decrease in vascular tone, which allows it simultaneously reduces that shear on the vascular wall and allows blood flow to increase to the target tissue. And this is mediated by nitric oxide, which is a potent vasodilator and prostaglandins in humans. There's also an independent vasodilatory pathway activated simultaneously that is composed of a variety of factors lumped into what we call endothelium-derived hyperpolarizing factors. But the goal is to dilate the vessel. So again, shear stress is on the vascular wall is a mechanical stimulus that results in vasodilation. An impairment of this mechanism is a hallmark of endothelial dysfunction, and this is a precursor and predictor in humans and animal models of atherosclerosis, which will lead to heart attack, stroke, and severe mortality outcomes. So cardiovascular risk factors in general, obesity included, impair flow-mediated vasodilation or flow-induced vasodilation. I'll be using those interchangeably in humans and animal rodents almost very, very robustly. And so this is likely through impairment of this signaling pathway here, the nitric oxide. However, again, upstream mechanisms and how this is being mediated are not well understood. Perhaps more perplexing and unique to obesity is that the dysfunction, and unlike other cardiovascular risk factors, the dysfunction in vascular beds is non-homogeneous. For instance, the visceral adipose that we could take from, say, the mesenteric adipose, these arteries exhibit robust endothelial dysfunction. Whereas if you were to take a more peripheral adipose tissue, like the subcutaneous adipose, and look at those arteries, they exhibit normal endothelial function. And so this is quite perplexing, and an easy question is why? Why is one vascular bed impaired and another seems spared? That's my little catchphrase for the day, impaired versus spared. And so this is evidenced by numerous groups. So this is pretty well established that there's this dichotomy that exists between these different vascular beds in an obese individual. And we see this with both receptor-mediated dilations and flow-induced vasodilation. So in this top panel here, you see subcutaneous arteries dilation in response to acetylcholine that are, these arteries are excised, pressurized, and preconstricted with something like endothelin-1, which then they can add acetylcholine in an increase in dose, and you can see an increase in dilation here. L-name is an inhibitor of eNOS, which produces nitric oxide, and you can see that that mechanism is intact in this artery type from an obese individual. So that's the subcutaneous nice dilations with intact nitric oxide signaling. In contrast, visceral arteries, again from the same individuals, have a very blunted dilatory response and no effect of blocking L-name, suggesting that perhaps L-name is, or eNOS and nitric oxide signaling are the impaired mechanisms here. The same is true if you look at a different agonist, the one that we'll be talking about most in terms of intraluminal flow and shear stress on the vessel wall. If you take those similar arteries and preconstrict them with something like endothelin-1 to induce a dilatory assessment, you put flow through the vessel, which is the shear stress. This subcutaneous arteries dilate very nicely. And papaverin is a control for the smooth muscle to make sure that in this individual smooth muscle is still intact, so we're isolating endothelial mechanism. In contrast, again, the visceral arteries do not respond as well to flow and are impaired. And what's not shown here is that this is also likely through nitric oxide impairment in this vessel bed versus subcutaneous arteries. So that brings us to a brief introduction of the titular ion channel. And so this inwardly rectifying potassium channel is where I'm investigating the role of this guy in these distinct vascular beds. And these are ion channels that in the monomer pass the membrane twice, so two membrane spanning domains, kind of a simple external moieties here and internal N and C termini. These come together in a tetramer to form the functional channel. So each different color ribbon here represents a single monomer that comes together and forms a functional channel. Now, the inwardly rectifying part of this, and this is important because you'll be seeing this again, these types of current profiles. So this is in patch clamp electrophysiology experiments, the way we can directly assess ion channels and their function. Current passes much more easily into these channels, hence the inwardly rectifying monomer here. And so current is going in. This is an outward current profile here, which is actually physiological. And so in these experiments, we can hijack the cell's membrane potential. We can ramp over time, changing potential, and look at current profiles. So this is actually measuring the channel itself, potassium flux, via this electrophysiology patch clamp technique. But the physiological portion, this outward current, is shown here. And so current in physiology, the potassium actually leaves and would hyperpolarize the cell and influence downstream signaling. But the currents are much larger inward and much easier to identify if there's changes. And so we'll be looking at these particular inward currents in our studies. And so what's the big deal about these ion channels? Well, it was shown all the way back in 1988 in a very famous Nature paper that if you can seed cells from culture into something where you can apply a flow or shear across them and record shear stress in real time, or the response to these currents to shear stress in real time, you can actually see that the currents, this downward deflection, is an increase in ion channel activity or current. And so that had a kind of dose-dependent effect on how much, so a higher dose of shear stress, so to speak, increased channels more so than a lower dose. And so you can plot that here. And so these channels are shear stress sensitive, which is important in the context of flow-induced vasodilation. And so the question becomes, why study these channels in obesity? And so we have, and I'll just describe some studies in language here, that we've showed previously that these channels are upstream of eNOS and nitric oxide in flow-induced vasodilation. So together, the fact that they're sensitive to shear stress and they're upstream of nitric oxide signaling, which is most likely the impaired mechanism in obesity, is kind of an important linker in terms of basic science to disease. We've also showed that a variety of other cardiovascular risk factors, including hypercholesterolemia, which is elevated cholesterol, impairs KIR2.1 channel function. So there's already an immediate link to other cardiovascular diseases or risk factors where the channel is no longer functional and is now contributing, its dysfunction contributes to vascular dysfunction. And perhaps most importantly, a study that we recently published was that the flow sensitivity of these clear channels is abolished in the visceral arteries endothelial cells of obese mice through a disrupted glycocalyx. We already have evidence that in obesity itself, in the impaired vascular bed, that this ion channel is dysfunctional due to the dysfunction of the glycocalyx. And so that's an important next step, is what is the glycocalyx and why is it interesting? The glycocalyx is also something that's been shown to be impaired in obesity. And what it is a series of core membrane proteins, glypicins and syndicins, that are adhered to these disaccharide sugar moieties called the glycosaminoglycans. And there's a variety of them, heparin sulfates, chondroitin sulfates, a whole bunch of sugars that kind of act as trees or leaves in the wind, if you, so to speak, if you consider blood flow as the wind, and these are kind of shaking and going. And if you get enough of a stimulus from that wind, then you can perturb the glycocalyx, and it will convert that mechanical stimulus into cell signaling and downstream mechanisms. And so it is pro-nitric oxide production. And in fact, if you degrade it, you can significantly reduce nitric oxide production. And so, as said, the glycocalyx is a Mechaner sensor, but also been shown to modulate ion channel activity. And so before we even kind of pursued the glycocalyx further, we wanted to be sure that the ion channel itself played a role in human flow-induced vasodilation. And so we also recruited subjects that were obese, that were undergoing planned bariatric surgery. We collected the subcutaneous and visceral adipose. We isolated the arteries from those adipose tissues, and then we exposed them to an adenovirus construct that contained a dominant negative KIR2.1 mutant. Now, this expression of this mutant downregulates the function of the channel. So it goes and it binds with all of the normal endogenous ion channels and renders them non-functional. So we can basically knock down the function of this ion channel specifically to assess its role in endothelial function in response to flow. And we do that by assessing the flow-induced vasodilation and pressure myography. And so what we showed is that indeed, in the subcutaneous adipose arteries, expressing that KIR2.1 exclusively in endothelial cells, that's important because it's driven by an endothelial cell-specific promoter, VE-cadherin, downregulates or blunts the response to flow compared to an empty virus control. And so from an obese individual, you get a nice dilatory response in the control that can be blunted when you downregulate the function of the channel. In contrast, an already blunted dilatory response to flow, which was expected in this artery type, the visceral adipose arteries from an obese subject, downregulating the channel has no effect. This falls in line with the lack of ability to block this response with nitric oxide, because the ion channel is upstream of nitric oxide. And so we think now we can infer that perhaps the ion channel is impaired, but this doesn't exactly tell us that directly. Okay, so there's a caveat here with investigating the obese human subjects. You're missing kind of the ability to investigate the lean control scenario. As you might imagine, you can't get, these individuals don't undergo bariatric surgery, so we're not afforded their full complement of taking the subcutaneous versus the visceral artery to compare them as we can in the obese scenario. So we can't do this. We're left with just doing in human work the obese subjects. And so what we did was we made use of a mouse model of diet-induced obesity. We were hoping it's a translational model of diet-induced obesity. And so what we did is we took mice in randomized groups at 10 weeks old and either maintained them on a normal chow for 8 weeks or more, or a Western high-fat diet to induce obesity. We wanted to see over time if we could recapitulate the human phenomena, where sub-Q would be retained function and visceral arteries would have reduced function. And after 4 weeks on diet, you can see that lean and obese, regardless of vessel type, respond just fine. Despite these animals gaining weight after a month on the diet, the arteries are just responding just fine. However, after an additional month on the diet, 8 weeks on the diet, the subcutaneous still the same, nice response to flow in either lean or obese arteries from lean or obese mice. In contrast, you now get to see a robust decrease in the response to flow after 8 weeks exclusively in the visceral arteries. And this maintained after 6 months on the diet, 24 weeks. Subcutaneous still just fine. However, visceral arteries from obese mice significantly blunted, but no further extent than 8 weeks. So we also wanted to look at smooth muscle function. We did this by applying sodium nitroprusside, which is an NO donor, so it dilates the smooth muscle directly using basically nitric oxide, and showed that at 8 weeks there's no difference between obese and lean arteries versus 24 weeks, where we get a significant decrease in arteries isolated from obese mice in the visceral. And this is just visceral to identify if smooth muscle dysfunction is occurring. And so this is the time point we use moving forward to isolate endothelial dysfunction from any smooth muscle dysfunction. This is the early time point we want to try and identify and determine mechanisms specifically of endothelial dysfunction in the absence of smooth muscle dysfunction. So is the ion channel also contributing this in this animal model the same way we saw in the human data? And the brief answer is yes. So after 8 weeks on the diet, we can significantly knock down the ion channel function or the response to flow by using the dominant negative. In barium, we use barium's an inhibitor of the ion channel, and so that was used as a control to make sure we're completely blunting the complement or the contribution of Tier 2.1 here. And so in arteries from visceral lean mice, so we have our lean controls here, the same is true. So in arteries from what you could say are healthy lean mice, the ion channel is contributing quite importantly to flow-induced vasodilation. On the flip side, subcutaneous arteries from obese mice still have this nice response and contribution of the ion channel to flow, whereas the arteries isolated from the visceral arteries isolated from obese mice no longer contribute as strongly, and there's no response of blocking with barium or the dominant negative here. And so blunted dilations overall, no role of the ion channel in FIV. So looks like there's an effect of obesity on this ion channel, but we wanted to test that directly. And so we have our patch clamp data with the inwardly rectifying currents from freshly isolated endothelial cells from lean mice here. And you can see that regardless of the vascular bed, you get nice response, robust shear activated currents. So this downward deflection in response to shear is an increase in currents, and this is the subtracted currents, nice, robust, shear-induced increases in KIR current. However, with obesity, we can observe decreases in baseline currents in a static bat in both artery types. However, in subcutaneous endothelial cells of an obese mice, there's still this nice response to flow. It's important to note that it's still significantly lower than this in the group data, but there's still a robust response in each cell type, which is abolished when we look at the ECs isolated from visceral arteries of obese mice. No flow response whatsoever in these ion channels, suggesting that perhaps that the saving of this response contributes to the preservation of the endothelial function in the gross physiology. Okay, so we're back to the glycocalyx. And what we can do here, and since we showed that the glycocalyx was impaired in visceral arteries of ECs of obese mice, so we already know that, we wanted to see if there was some role in the subcutaneous as well. And so how we do this is we use atomic force microscopy with a lever that approaches a specialized functional probe that approaches the glycocalyx. And it touches the glycocalyx and continues down, and we can get force over indentation depth here. So we can, from that, we can extrapolate stiffness of this glycocalyx, so how much force and resistance is present here. And we can also get the thickness of the glycocalyx from this information. And so that would tell us if there's less of this stuff here, that it would be degraded perhaps, as well as stiffer. A shorter glycocalyx, you might infer, would be stiffer. And that would be a marker of dysfunction. So the question is the glycocalyx only altered in visceral adipose arteries? That being said, we think that it would not be as affected in subcutaneous endothelium because those ion channels were still functional, and there's a relationship between the glycocalyx and Kehr channel function. However, what we see is that there is a significant effect of obesity on the subcutaneous artery adipose glycocalyx in terms of stiffness, However, it is much more pronounced in the visceral adipose arteries. The glycocalyx is much stiffer compared to a lean animal. And interestingly, when you take these and compare them within a diet type across arteries, there is no significant difference in the glycocalyx stiffness when you compare the artery types in a lean mouse. However, in the obese mouse, the visceral adipose artery glycocalyx is much stiffer. Similar findings occurred when we look at the thickness of the glycocalyx. So there is a significant decrease in the thickness of the glycocalyx with the subcutaneous adipose arteries. However, this is much more pronounced when we look at both the between diet as well as the within diet comparisons of artery types. So this suggests that perhaps the mild disruption of the glycocalyx and subcutaneous adipose arteries results in a mild decrease in the currents, the flow-induced currents, while this much more exacerbated phenomenon in visceral arteries results in an abolished tier 2.1 response to flow, which results in endothelial dysfunction. So the future directions here all pertain to the mechanisms driving this glycocalyx dysfunction, and the first would be role of adipose depots in disrupting the glycocalyx and inducing endothelial dysfunction. Is there a paracrine mechanism mediated by the spatially distinct adipose for which these arteries are embedded? So is there something going on in a sick visceral fat that is inducing impairment of the glycocalyx, which results in dysfunction of the ion channel and ultimately endothelial dysfunction that is not observed in the subcutaneous adipose? And we presently have RNA sequencing data and analysis from the respective cores to actually try and find some interesting targets that might be mediating these mechanisms. We're also interested in performing some fat swapping experiments that we can do to induce dysfunction, to see if we can switch or induce dysfunction using visceral fat on otherwise healthy arteries, or can we recover visceral artery dysfunction by incubating with, say, healthier fat from lean mice or humans. We also would like to do some intervention studies and maybe working with Christiana or bariatric surgery units in the area so that we can maybe manipulate diet or exercise in humans and in animal models to see if we can, in an obese environment, preserve these through interventions, preserve endothelial function specifically in the visceral arteries. There's also some basic science questions that pertain to how does the glycocalyx activate shear channels in response to shear stress. So is there a tethering mechanism that's a physical connection between the glycocalyx gags or the internal structures that would transduce the direct response? And this is in collaboration with Irena Levitin at UIC. So that's what I have for today. I'd like to thank you all for listening, and I would like to point note to my UD group here, the UIC collaborators, as well as some funding sources, especially the COBRI that I'm currently attached to here at the University of Delaware. Thank you.